Thank you for those kind and largely undeserved words. As we know, those of us who are in the academic world, now we put advertising breaks into our lectures. Here it is. Here's one of my books. It's called The Single Helix. And it's called The Single Helix for two rather witless reasons. One of which is the Latin name of the snail I have worked on, is Helix. And of course, the other which, the most famous book in science, in popular science without question, is called The Double Helix. And as I said to my publishers, I don't mind if my book is only half as good as The Double Helix, as long as it sells half as many copies, but unfortunately it shows no signs whatsoever of doing that. There is, of course, a message in the single helix and indeed in the double helix. And here is the genotype, the DNA, a bit of the DNA of of the man who wrote the double helix, Jim Watson, who famously, and that's rather blurred, but most things that come out of Jim Watson are rather blurred nowadays. Jim has displayed his entire DNA for public view, apart from the one that might predispose him to Alzheimer's disease, which he's kept quiet to himself, which of course he's entitled to. And it really makes a statement of the astonishing progress which has been made in the technology of genetics. We're now talking about the $1,000 genome, certainly the $10,000 genome, possibly a genome that can be sequenced within a day. Hasn't come yet, but it might come. And many people think that actually this is going to revolutionize the way that we look at ourselves, the way that we practice medicine, and much more. And I'm not going to talk about that, or I'm not going to talk about it scarcely at all. Instead, I'm going to talk about the single helix, about snails. There's a good general rule if you work on snails, and I still do. Nobody ever becomes famous until they stop working on snails. And there are some surprising examples of that. Here's a book, The Conchologist's First Book. The conchologist is somebody who works on snail shells, okay? It sold large numbers of copies in the 19th century. If you read down its rather lengthy title page, you've got a rather surprising author at the bottom, a chap called Edgar A. Poe, Edgar Allan Poe. That was Edgar Allan Poe's first book, which outsold most of his fiction in his lifetime. I wonder what happened to Edgar Allan Poe. Okay, here's another one. Here's Lewis Carroll. And Lewis Carroll, famous for many of his interests, one of which is illustrated here, was very much a naturalist in the 19th century tradition, greatly interested in snails, published scientific papers on snails, more famous for discovering new kinds of animals, such as this one, beware the jabberwock, beware the jubjub bird, and shun the frumious bandersnatch. I have identified what the bandersnatch actually is, because if you look at papers in German about the snail I work on, Helix hortensis, Helix normalis, by den Banderschnecken. So the bandersnatch is in fact an enormous rabid mollusk, you might like to know. All right? So again, he's given up working on snails and he's become famous. I carry on and remain and stay infamous, and I'm in the middle of giving my introductory lectures to first-year undergraduates at UCL. 24 lectures. I start them off by saying, I'm a geneticist and my job is to make sex boring. And after 24 lectures, they certainly agree with me. And after that rather pathetic little joke, I show the seriousness of the subject by asking them, as I'll ask you, to look at the person to the left and the person to their right. And I say to them very seriously, I can assure you that 2 out of every 3 of you will die for reasons connected with the genes that you carry. And they look a bit baffled at that, and I say, well, cheer up. If I've been giving this lecture in Shakespeare's time, 2 out of every 3 of you will be dead already. And of course, that's true. And here are the patterns of life expectancy of English babies. From 1600, about 1 in 3 lasted to be 21. In Dickensian times, about half. And now about 99% of all British babies English babies live to be 21 years old. However, they all die in the end, of course. We all die in the end, of course. And what kills us tends to be not the enemy from outside, cholera, starvation, cold, but the enemy within, as Mrs. Thatcher might have put it, things like diabetes, cancer, and heart disease, many of which have a strong inherited component. And actually, I wish I was around, which I probably won't be in 100 years from now, because I could add another 2101, because I could probably add another line to that table which would show a drop in life expectancy for a reason which I'm sure you're all familiar with, which is the spread of a great plague, which certainly has some genetic component, the great plague called obesity, an illness which is going to kill lots and lots of people off, and which there's a huge amount of genetic interest in. And here's the patterns of obesity with time from the 1970s plotted up to 2030, and we get to 40 or 50% obese really quite soon. So that's a problem. A lot of genetics in that. Here are some genetics of obesity genes, but I only mention that to show the way in which the study of DNA has actually moved into things that we might think to be public health issues, into things in this case of obesity. Now, what's completely revolutionized genetics, of course, is technology. We all know about DNA. If any one of you, bored stupid by my talk, was to rush out into Kensington Gore and be squashed flat by a number 10 bus, the DNA in your body would stretch to the moon and back 8,000 times. That's a lot of DNA, okay? Many, many millions of miles of DNA. And famously, it has a rather small number of genes, about 20,000, about the same number of moving parts, in fact, same number of parts as in a number 10 bus. So, you know, we're not much more complicated than a double-decker bus, which is quite surprising, really. But one of the interesting aspects of genetics, which is often forgotten, particularly by molecular biologists, is what genetics all about. It's about difference. And if you look at people who sequence DNA, the original DNA sequencers, which is a wonderful piece of work, of course, and Jim Watson's own DNA sequence, it's a very platonic view of what life's all about. You've got a human being, an example, an exemplar, J.D. Watson. You've got his DNA. That's the end of the story. But of course, it isn't like that. If there wasn't difference, then we wouldn't have genetics. We wouldn't have evolution. We'd all be stuck in the primeval slime. So genetics has moved on to begin to ask questions about difference. Why are people, why are snails so different from each other? And actually, we're probably more different than you think. Another rather silly and perhaps slightly childish experiment is to ask each one of you, and this works well with 8-year-olds, I find, ask each one of you to stick your tongues out at your biology teacher, but you can do it at me, could do it to me. Stick your tongue out at me and try and roll it up at the sides. I can't do it. Who can? And who cannot? And that's actually, to some degree, and the geneticists in the audience, I apologize for this, to some degree, that represents a genetic difference. Here's another one, perhaps less familiar. Without thinking, that should be easy, clasp your hands together, and, you know, England won the rugby, right? Okay. Who's got the left thumb on top? Who's got the right thumb on top? Try it the other way. It's very weird. And again, there's a heritability of that as well. So there's some genetics in that. And that's a statement of difference. And that's what I'm interested in. How did differences get there? Why are they there? And how many are there? Certainly, if you look at the human genome sequence, and here's a little bit of it, this is not going to be in the exam, I assure you. It looks boring. But now we know that it's actually fascinating because there's a huge amount of difference in it. In the last few weeks, image of it has changed completely as technology has gone on. They're far more different than we thought before. Here, for example, is a diagram that shows the accumulation of what we know about single letter differences in the code from 2000 to 2005. 2005 was prehistory in this story. In 2005, we had 8 or 9 million sites in everyone's DNA that potentially could vary from person to person. That number's now gone up to about 30 million. So everybody in this room is different from everybody else, and also different from everybody who ever has lived or ever will live in the future. And a lot of those differences are going to make have a large effect on your future. So the question is, why are they there? It's very hard to know with humans. There's a limit to things, what you can do to human beings. They're not ideal experimental animals for geneticists. They live as long as a scientist does, and you can't set up matings with any great ease, although humans, I'm a great believer in the healing power of lust. Most matings have been done. So a lot of things you can't do with humans, but you can do them, guess what, with helix, with snails. And that's what I want to continue to talk about. Here's the thing about snails, and the snail I work on, is that they're fantastically variable, okay? What I would like you to do is just grab some of these animals. You can make jokes about shell shock if you like. And you can say, when that joke doesn't work, you can say, this one comes from a broken home, all right? Okay? Now, what you might do is pick up one or two of those snails and just compare them with each other. And I've got some more, which I'll cease throwing around. And they're rather battered old snails, these. But if you compare them, you'll find, and they're all the same species, you'll find that they differ from each other in the color, or the color's faded for some, in the number of stripes they've got, whether they've got a white or a dark lip, and so on. And this is difference. And people have been working on these snails for 100 years or so to try and find out why they're so different. Well, I've wasted nearly 40 years in doing this, and I won't bore you with the full story. But one of the reasons, clearly, why they vary in color from place to place is actually that the dark and the light, or the stripy ones and the non-stripy ones, soak up energy from sunshine to differing degrees. Some of them, if they're dark in color, soak up more solar energy. It's physics, got to be true. Some of them, if they're light in color, reflect it and soak up less. And indeed, if you map out the genes across the world, you get a very clear pattern, almost as clear as in humans, of dark color in the north and in cold places, and light color in the south and in warm places, okay? Humans, of course, are the other way around. Well, that's interesting, but I'm not going to go into why. The more interesting question is, why does any single population, as it nearly always does, contain both dark and light snail shells? I do a lot of my work. There's the snails. They're slightly better, slightly nicer state than the ones you've got. They live in interesting places. Here's a Dutch flower painting. And if you look carefully, you'll find two snails, don't show up too well there, well hidden in quite a complicated environment. I do most of my work in the Pyrenees. These snails only live in national parks, I'm glad to report. And we've done a lot of work there. I've just come back from there, in fact. As I say, most populations are variable. Why are they variable? That's the interesting question. There's a sample you might get from a population with both dark and light in it. Well, why? Let me skip over the next little bit. Pity about this. These balls are called Jones's balls for reasons you'll never know. Okay, I'll skip over that. What you need to know, well, the interesting thing is you've got a lot of variation in there, in a population living in a hedgerow. Why aren't they all dark or light? Could it be that within this few square meters of habitat, there is enough patchiness, enough variation in the environment to allow both types to survive? What you need to know is, are these animals choosing to live in different places within that little patch? Well, you can watch them, and we have watched them, and it's not really much fun. They do this now and again, and that's about it. They're not very active, they're sluggish. Another joke about snails. What you need to do is to assess how much time each one has spent in sunlight. Armand Leroy, I don't know, he's probably stormed out now. Is Armand around? If you look at Armand Leroy's trousers, okay, you will see he's probably the last person in the Western world still to be wearing jeans. Well, okay, and he is definitely the last person in the Western world to be wearing faded blue jeans. 20 years ago, I wore faded blue jeans. I've grown out of it, Armand. And I was looking at my jeans one day, and I thought, hang on a minute. These jeans are faded because the pigment they're colored with, blue, is faded in sunlight. Can't I do a bit of, weak joke coming, gene manipulation, cut squares of this stuff out, and stick it onto snail shells and see if we can tell how much time they're spent in the sun? Well, it didn't work. It was ridiculous. But what we then did was to take the blue pigment, which is used to color genes, mix it with yellow paint, which makes a green paint, put the spots on snails, and some of the snails you've got will have those spots on them, and put them out in the wild, and come back and collect them a month later, and this is what happens. Some individuals, the dark green on the left there, have scarcely been exposed to the sun, and some on the right have spent a lot of time in the sun. And if you look at dark and light-colored snails, genetically distinct, unbanded and banded, as I say, within a population, these individuals spend different amounts of time in sunlight. So that's a statement that somehow genes are choosing habitats. Over a tiny distance, a square meter, different individual snails are choosing to experience a different way of life, which I find interesting, and we published in Nature, had absolutely no impact whatsoever as far as I'm aware. We then went on to an early piece of gene manipulation, which involved black paint. We took light-colored ones and painted them black, and black-colored ones and painted them white, did the experiment again, dark-colored ones and painted them white, did the experiment again, and their behavior switched. So it was a straightforward mechanical kind of thing. Well, let me end up, that's interesting, I think, to a population geneticist, because one of the embarrassments in population genetics is that we do not know why variation is there. And it may be, even in the human context, that people with different genes, different abilities to digest fats, let's say, certainly different ability to drink milk as adults, they may choose different diets as a result. And this is a new aspect of human genetics, which is really getting going now, although the single helix people perhaps got in there first. Of course, we have actually, just to end up, I did play with this technology with humans. I spent some time working in Botswana in southern Africa many years ago with sand people. I had sand students who didn't sing like this. They didn't sing like that. They sang nearer to God, nearer my God to thee most of the time. They were very strongly Christian. But one of the issues in southern Africa, and indeed in West Africa, is that there's a surprisingly high incidence of albinos, African peoples, without any skin pigment. And that's quite a big, we don't know why, it's a mystery, but that's quite a big medical problem, because these children in particular, if they play in the sun, of course, as they would like to, they tend to get skin cancer, which kills them off, later in life. And a colleague and friend of mine, Trevor Jenkins, from the University of Witwatersrand, was interested in trying to change their behavior, explain to the parents that these kids should not spend time outside unless they were wearing hats and long sleeves. And so he set up this little experiment with the green paint. Actually, we used it to dye cloth, where we made them hats which are actually made of pigment which faded, made of a cloth which faded in sunlight, and little badges which they wore which said, "I am in the experiment." And the idea was that you would allow them to go out and play for a week or two, and you compare the hat and the badge to see how long they've been wearing their hats, because they tell you, of course, that they've been wearing their hats all the time. But of course, like all 8 or 10-year-old kids, they'd be telling you what you wanted to hear. And the idea was you'd hope that the hat and the badge would fade at the same rate, so you could see how much time they'd worn the hat. It didn't work, because these kids, being extremely charming and helpful, they always put the hat on the windowsill to make sure it faded, and we got the result what we wanted. So that's really a statement that actually when you're doing human genetics, you have to realize that you're dealing with people. When you're dealing with snail genetics or fruit fly genetics, you're dealing with snails that don't have much common sense. That's particularly true, perhaps, when you're looking at the genetics of obesity. Let me just turn back to that, which has really become almost an industry now. Here's a slide I show as the very end of my 24-lecture college genetics course to first-year students. It's about the genetics of obesity. And here's a family, sadly familiar image perhaps, in an American context, but certainly to be seen here in Europe, 2 overweight obese parents and their obese child, and we can talk about the inheritance of obesity and the genetics which might be behind it. But I always end my course by showing a picture of this obese family's cat, which looks like that. And that really makes a statement to you that actually there is a great limit to what genetics can tell you about what we ourselves are. I will stop there. Thank you.